Dr. Allen. I think we're going to go ahead and get started here. All right, yeah. It's my pleasure today to introduce our speaker, Dr. Masfique Mehedi from the National Institute of Allergy and Infectious Disease. Dr. Mehedi received his Ph.D. from the University of Manitoba in the laboratory of Heinz Feldman, where he studied transacting factors involved in RNA editing of Ebola viruses. After leaving Manitoba, he joined the laboratory of Peter Collins at NIH, where he has been studying human respiratory syncytial virus host factors, which we're going to hear a lot about today, I assume. Dr. Mehedi's published his work in top journals, including PNAS, PLOS Pathogens, the Journal of Virology, and many others. He's also received numerous awards for both his research and student mentoring capabilities, which I thought was very nice and speaks well. The title of his talk today is Host Factors for Human Respiratory Syncytial Virus Cell-to-Cell Spread. Please join me in welcoming Dr. Mehedi. So, welcome.
Thank you, Dr. Archer, for kind introduction. It is truly an honor for me to talk about my research in front of you. The title of my talk is Host Factors for Human Respiratory Syncytial Virus Cell-to-Cell Spread. I begin with telling you my journey of learning. I'm from Bangladesh. I completed my BSc as well as master's back home. During that time, there were frequent outbreaks of Nipah virus. Therefore, I wanted to study Nipah virus. And that's why I went to Canada to finish my Ph.D. under Dr. Heinz Feldman. Instead of doing work on Nipah virus, I ended up working on Ebola virus. During my Ph.D., I got a fellowship to work in the BSL-4 laboratory at Rocky Mountain Laboratories. And after finishing my PhD, I joined Dr. Peter Collins' lab to work on respiratory syncytial virus. Today, my talk will focus on respiratory syncytial virus, the work I have been doing under Dr. Peter Collins. In my talk, I will give you a brief background on RSV. I will show you how a genome-wide siRNA screening can be helpful to identify host factors. I hope to convince you that I identified a novel host factor for RSV, as well as identified a novel RSV spread mechanism. I'll wrap up my talk giving some future directions of my current research. First, RSV. RSV belongs to a new virus family, Pneumoviridae, genus Octopneumovirus, and a species human respiratory syncytial virus. My talk is primarily focusing on these species. RSV can be an envelope virus, and virion can be paleomorphic form. One of the examples is filamentous RSV virion, and this virion contains a non-segmented single-stranded RNA genome. RSV genomes contains 10 genes encoding for 11 proteins. One of the most important proteins is fission protein F, which is a surface glycoprotein, has a major role in virus entry and cell-to-cell fusion. In RSV, infected cells making a multinucleated cells called syncytia, which is a hallmark of RSV infection in vitro model. Why do we care about RSV? Because RSV is the leading cause of acute bronchiolitis, and it is one of the common causes of infant viral death worldwide. Most children get RSV by the time they're 2 years old, and its prevalence is worldwide. As you can see, nearly 34 million RSV-associated cases worldwide annually, and 10% of them require hospitalization. And there are reports of 200,000 deaths. Unfortunately, 99% of them in the developing countries. RSV can cause disease in elderly and in transplant recipients, as well as immunocompromised patients. But there is no vaccine or routine therapeutic antiviral drug available.
The audio is not coming through. Just want to make sure everybody can hear you.
Okay. Can you hear me now? And as there is no approved vaccine or routine therapeutic drug against RSV, it is a global health and economic burden. And my research is working on host pathogen interaction to identify therapeutic targets to combat RSV infection. Another reason is we've
less we know about the host factors involved in the RSV infections. And to study host pathogen interaction, I use siRNA-driven gene silencing. And we know that siRNA has been well known to bind to the specific mRNA by using RNA-inducing silencing complex to chop out the mRNA that will produce the protein. So in that way, using siRNA, we can block or deplete protein expression in a cell. So here is example of one single siRNA. In my study, used a genome-wide siRNA screening. So that means it's a high throughput. We used a human lung epithelial cells, which is most widely used in RSV research. And for the screening, we used RSV virus that express green fluorescent protein that allows in the high throughput context. We also use commercially available silencer select genome siRNA library. And screening has been done in collaboration with the National Center for Advancing Translational Science, NCATS. Here is a schematic diagram how we done the siRNA screening. We screened nearly 22,000 genes by using 3 independent siRNAs per gene. You can imagine that there are 66,000 siRNAs we used. Basically, we used 1 siRNA per well, and then we used A549 for incubate for 48 hours to allowing enough time that siRNA-driven knockdown of a particular protein will be achieved. And we infected those cells with RSV GFP using multiplicity of infection of 1. And 48-hour post-infection, we fix those cells and stain for nuclei to check the viability of that cells. And we also quantify GFP signals. Here's an example. We used positive, here, when we use siRNA targeting GFP, you can see substantial reduction of GFP expression compared to siRNA control. And based on the nuclear staining, we can see the viability of the cells. In the siRNA, we call as a hit when 2 of the 3 siRNAs showed 50% reduction of the GFP expression, but the viability did not reduce more than 50%. So in that way, we plotted all the screening data and follow the normal distribution pattern. And we can identify candidate genes based on 2 or 3 standard deviation from the sample mean. And even though we used a very stringent criteria to selecting candidate, siRNA screening has an inherent problem or off-target effect. Off-target effect is when a siRNA bind complementary to the target mRNA, then we can expect that the single intended RNA will be targeted and will be chopped out. But if it is partial complementarity, and then the single siRNA can partially bind in multiple mRNAs, and then multiple unintended target can be reduced, and therefore the bias in the screening can be achieved. To overcome this off-target effect, we used a common seed analysis. So here's an example how we can distinguish between on versus off-target in our screening result. For example, SCAMP5, for target this gene, we use 3 independent siRNAs. And siRNA has their seed sequence that allows siRNA to bind. And you can see 2 of the siRNA out of 3 showed 50% reduction in the activity compared to the screening median. But if you look closely, that other siRNAs also have the same seed sequence showing that similar reduction. What it suggests that these are the seed bias and most likely false positive. So another example we want to show you that XRN1, and again the 3 independent siRNAs and their seed sequence here. Based on their activity, we can see a substantial reduction of the GFP expression when we use individual siRNA. When we checked the seed sequence of other siRNAs, their result is close to the screen median, suggesting that this is truly positive. And by this way, we can exclude off-target effect. And from, off-target effect from 22,000 candidates, we narrowed down to 155 based on the ranking. And these 155 were re-screened with additional 3 independent siRNAs from another company. So therefore, the 6 155 candidates we screened with 6 independent siRNAs. And eventually we narrowed down to 65 potential candidates for validation or further characterization. So the way we did I beg your pardon, it's too touchy here. So what we use, we exactly use the similar high throughput screening context, but here we used a 24-well plate, allowing enough cells to give the results more validation of what we see on the high throughput context. So basically, we exactly used the similar strategy, individual siRNA per gene, and then we transfect for 48 hours, and we infect those cells with RSV-GFP for additional 48 hours, and we checked for cell viability based on Alamar blue staining. We checked for viral protein expression by quantifying GFP signals, and we also determined virus production using immunoplaque assay. Here I'm going to show you the results of all the 65 genes. I plotted from left to right based on RSV titer, highest to lowest, and the red bar representing the viability of those cells in that well. And overall, due to siRNA knockdown, the viability did not compromise much. And in those well, you can see a majority of them showing at least 50% reduction of the virus titer. And virus titer reduction also in some cases complement with the GFP production, but not always the case, because in some of the examples you can see virus titer reduced, but the protein expression did not reduce. But overall, the majority of the knockdown showed that at least 50% virus titer reduced, suggesting that these 65 potential candidates could be associated with RSV infections. To characterize 65 is a big number. I have to narrow it down to some candidates that can be achievable for characterization. For that, I could have chosen candidates with highest virus titer reduction, or I could have chosen candidates with highest GFP signal reduction. Or third option is using an ingenuity pathway analysis, putting all the candidate gene list in the knowledge base software, and then identify canonical pathways most likely involved in the virus infection. I choose the third option. When I use 150 candidates in IPA to look for canonical pathways, I found that top 5 canonical pathways I showed here. Integrin signaling is the topmost likelihood association with the RSV infections. And interestingly enough, 4 of the 65 candidates are associated with integrin signalings. So now from 65, I have 4. And to know about the integrin signalings can be involved in the cell movement or organization of the cytoskeleton. And these 4 proteins are actin-related protein 2, ADP ribosomal factor 1, integrin alpha 5, and mitogen-activated protein kinase 3. And to elaborate in another pictorial diagram, I can expect that integrin should be in the plasma membrane when ARF1 is known as a transport protein from ER to Golgi transport pathway. MAP kinase is a cellular kinase, can be involved in different cellular processes, and ARP2 is an actin polymerization protein, allows actin polymerization. Now I'm going to show you like all these protein characterization, whether these are actually involved in the virus infection. First, ARP2. So what do we know about ARP2 is it is a major constituent of ARP2-3 complex. And this complex has 2 major proteins, ARP2 and ARP3, and 5 cofactor proteins. This ARP2-3 complex allows globular actin to form follicular actin. And this process is also known as actin nucleation. This actin nucleation or actin polymerization allows to form Filopodia and Lamellipodia. And these Filopodia and Lamellipodia has been shown to involve in cell shape and motility. So first thing I wanted to investigate whether ARP2 actually involved in virus infection process. I was able to identify a single siRNA that successfully knocked down ARP2 expression in F549 cells and did not reduce the cell viability. So first thing I tested whether ARP2 involved in virus entry process. And for that, in the knockdown cells, I infected RSV GFP and quantified the number of infected cells. At 16-hour post-infection, knockdown or without knockdown, the number of infected cells was almost equal, suggesting that ARP2 may not be necessary in the virus entry process. We confirmed that results with when we use a constitutively ARP2 knockdown A549 cells as well. And we also used an inhibitor targeting ARP2/3 complex known as CK666. We also used EIPA as a positive control because EIPA is a macropinocytosis inhibitor, has been shown reduce RSV entry. And in our results, we can see that EIPA reduced 60% of RSV entry when in the same concentration CK666 did not reduce RSV entry process, either 6 hours or 12-hour post-infections, confirming that ARP2 not necessarily involved in RSV entry process. Then we asked whether ARP2 is involved in the virus transcription or translation process. And for that, we used qRT-PCR quantifying RSV mRNAs. I checked every single mRNAs from the RSV genome during transcription. But here, for example, I'm showing fmRNA. You can see over time, total mRNA is reduced due to ARP2 knockdown compared with siRNA control. And if it is mRNA reduced, we expect that protein should be reduced as well. Indeed, we found that at the later time point, the reduction of the RSV protein was achieved. And also another protein, RSV P protein, also showed the similar indication. But it is the total protein level reduction due to ARP2 knockdown. We also wanted to know this protein reduction is whether it is due to the spread or not. Because when we quantified the number of infected cells quantified by the different independent individual RSV protein, by FACS analysis, we found that 24-hour post-infection number of infected cells are almost similar. And for 48 hours post-infection, this number of infected cells reduced by 50%. When we compared the mean fluorescent intensity for each of these proteins between 24 and 48 hour post-infection, we did not see any change. What it suggests that this 50% reduction of number of infected cells is due to the spread. Because if virus can infect cells, the protein expression did not change due to ARP2 knockdown. Rather, number of infected cells is reduced. What it suggests that ARP2 may be important in the virus spread process. And if it is true, then we can expect that virus production will be reduced. Indeed, we found that. When we quantified total virus titer in the supernatant, we found that there is a 10-fold reduction of RSV production due to ARP2 knockdown. We are able to recapitulate this result with another respiratory epithelial cells, CALU3. And I'm not showing here, but we also used verocells, or standard cell line used in the virology research. We also found that the similar results. What it suggests that we were able to identify ARP2 effect in virus production through independent cell line, but in more likelihood is involved in the RSV spread process. We wanted to investigate further whether our ARP2 is involved in the virus morphology or budding process. And to do that, we used electron microscopy, basically transmission electron microscopy, to when we check the infected cells and we look for the virion production. And we found that regardless of ARP2 knockdown or not, the filamentous virion we observed whenever we checked. So it suggests that ARP2 may not be necessarily important for RSV virion morphology. When we checked budding process, for that we used in a different electron microscopy strategy. Basically, it's a scanning electron. I beg your pardon. Yes. The scanning electron microscopy. So what we found that here you can see, let me walk you through the graph. The A549 mock cells is our control, and the infected cells showing that virion laying on the surface. And these we can call virion because of the length is 2 to 10 micrometer in length, and also 100 nanometer wide. And these are virion we also confirmed by the RSBF protein we detect using immunoglobulin antibody. So we confirm that these are the virion. In contrast, ARP2 knockdown cells, we did not see virion laying as like siRNA control. Rather, we see that irregular virion budding, and in some cases perturbation of the RSB budding because of the cluster of F antibody, and it's completely different compared to siRNA control. So therefore, we also wanted to investigate what is the, because this is a magnification of, we want to use the lower magnification to see the overall view, how the ARP2 knockdown cells looks like under scanning electron microscope. What we found that mock cells usually use the extracellular extension, and ARP2 knockdown without infection, you can see the extracellular extension was not abolished or reduced. And RSV infection increased dramatically the extracellular extension extensively, and every single cell is showing that extracellular extension. And this extension looks like Filopodia or Lamellipodia. That we'll investigate further later. But another observation was that ARP2 knockdown, with this extracellular extension was abolished or reduced substantially. And to investigate whether actually these are the Filopodia or not, we use confocal microscopy.
So I'm curious, after infection the cell, is the cell still healthy, the proliferation ability compared with the infection?
Yes.
Yeah, my question is after infection with the ISV, your A49 cell is still very healthy, their proliferation ability all the same?
Yes, proliferation ability it depends on the time point. If we keep longer, the virus infection will have a cytopathic effect. So your question was whether RSV infection increase cellular proliferation. My understanding is RSV infection taking advantage of cellular machinery for their benefit. As a result, cells may not be as efficient in respect to proliferation. And as a virological standpoint, that RSV infection has a cytopathic effect. Those cells will be getting die over time.
So what's the time limit? How long after infection will be affected? What do you test for that?
Yeah, the question is how long we can infecting cells can be persist. So it also depending on how much virus we put to start with. And at the same time, to answer your questions, is that RSV has a tendency to regulate their cell death pathway to allow us to stay longer for their benefit. But in the in vitro model, if we put less virus, then it will persist longer. And the day 3 post-infection, you can see all the cells will be trash.
So then the final question is a match to your, you say that the mechanistic 3 also changes, right? You focus on them, you listed mechanistic 3 also changes. Is it consistent, rapid change, consistent mechanistic change, the pattern is consistent or not?
Yes, in respect to MAP kinase, it was a part of the integrin signaling pathway. So the way our project went on is first we wanted to characterize ARP2 effect, and what we found that ARP2, in addition to ARP2, other factors like the MAP kinase as a part of integrin signaling, we were able to see that that virus production reduced. But to our surprise, that I will show you that results, that MAP kinase effect is very less compared to ARP2, ADP ribosomal factor 1, and integrin alpha 5. So MAP kinase is in respect to, if we do knock down MAP kinase 3, you do not see much reduction of our RSV production. But as a component of integrin signaling pathway, we do see there is an effect on that. But the effect is very less or negligible, and depending on the study we are looking at. because I will show you, if you allow me, I'll come into that point in the later of my presentation. And I beg your pardon, the clicker is a little bit sensitive. I hope you will bear with me. So to investigate further whether these are the Filopodia or not, I used confocal microscopy. And what we found that RSV infection extensively induced Filopodia. And these are Filopodia, we can call it, because it is similar to growth cone, neuronal growth cone. And as you can see, the microtubule, it is not necessarily into the Filopodia. And we also found that Filopodia is microtubulin deficient. So we call it as a Filopodia based on their size, length, as well as microtubule deficient. So what we found that if we do knock down ARP2 and infected those cells with RSV wild type, we, to our surprise, that the Filopodia is abolished or reduced, but the cell is a heterogeneity. So therefore, we want to quantify number of independent experiments, and I plotted the data in a combine. As you can see, RSV infection increase the length of the Filopodia, and length can be more than 50 micrometer, more than that. And ARP2 knockdown not only reduced the number of Filopodia, but also the length of the Filopodium as well. So we were able to identify that RSV induced Filopodia-like structure that is completely novel, that we didn't know before, and ARP2 as host factors in that process. So I try to make a simple diagram. What we found that RSV entry did not impact by the ARP2, rather RSV infection induced Filopodia and ARP2 involved in that process. So we wanted to know whether Filopodia has some impact on the virus pathogenesis. And what we found that other pathogens already using ARP2-3 complex activities for their benefit. Example, E. coli, HSV1, vaccinia virus, and a list of pathogens you will find they use for their cell-to-cell spread, as well as cytosolic movement, or in some cases phagocytosis as well. So it's not completely new. Rather, for RSV case, it's completely new that we found that. We wanted to investigate whether this Filopodia is part of or associated in the RSV replicative cycle. And for that, we used a high resolution stimulated emission depletion microscopy, where we found that virion can be budded from Filopodia. And Marburg virus has already been shown that primarily budded from Filopodia. So in our case, RSV also budded from Filopodia. And we also wanted to know whether this Filopodia can reach out to the neighboring uninfected cells to transmit virus. And interestingly enough, we found that RSV use Filopodia to transfer from one cell to another. Here is a good example. There are 3 independent cells, A and C. Hope you'll appreciate that these are highly infected, and the B is immediate infection. And if you see follow through the Filopodia length from cell A to cell B, at the touching stage, all these virion filaments are there, suggesting that these virions coming from uninfected cells to uninfected cells. So this is for the first time we're able to see how a cellular extension used from virus spread from cell to cell. And ARP2 is involved in this process, then we would expect that this process will be impaired. And exactly true. Or when we used a constitutively ARP2 knockdown F549 cells, and we identify that not only filopodial induction was reduced, but also cell-to-cell spread was not obvious due to ARP2 knockdown. So confirming that RSV infection induced filopodia and filopodia used in cell-to-cell spread. We used as a fixed cells. We wanted to visualize this process. And to visualize that, we used a live cell imaging using the confocal microscopy. We imaged over time and make a movie. So here we used the red affecting cells, and these cells, RSV GFP we used, we expect that these cells, if in fact this cell, then these cells will be gradually green. And when we, over time, when we found that, as you can see that it's transmitting virus, and you also, you're able to see that these two cells are transmitted from these infected cells. And if it is true, then these two cells will be gradually green. And exactly over time, it gets greener. And one thing you hope you'll appreciate that infected cells is more dynamic. And as soon as they are dynamic, they're making Filopodia-like structure as well and trying to reach out other cells to transmit that virus. So that is quite amazing that infected cells getting more dynamic and try to spread virus using the cellular extensions. And if our, here is a good example. Also, you can see how extensive the Filopodia extension could be. And if ARP2 is involved in this process, yes.
It's a fantastic video. Is this something that's unique to A549 cells, or did you try the same? Did you see this in other cell lines outside of that?
Right. The question is whether this Filopodia is unique to A549 cells or not. Answer is yes. In respect to compared with another cell types is the monkey kidney varicose line. And I will talk about my future project. Like I see that in the lung epithelial cells, when varicose cells is a monkey kidney epithelial cells. So one of the interesting thing I want to ask the question whether there is a cell type specificity. But to establish that, I need to reach out or need to investigate additional cells like the nasal epithelial, some other respiratory epithelial cells to establish that. So that is my future project. No, not extensively, not extensively.
You said this, but what's the derivation of the A549 cells? What is the derivation? Where do they come from?
Yes.
They're continuous cell line.
Correct.
And
they're from the lung epithelial cells. It's a type of pneumocyte cells.
Okay.
And it's come from the alveolar epithelial.
Human?
Yes, human epithelial cells.
They're the alveolar type 2.
Yeah.
Yeah, they're adenocarcinemic.
Ah, that's the transformation. Okay.
Yeah, and if I allow me to, I believe the cell polarity is another factors in this process may play a role, but I use this 2D culture system. We may not be, say, completely polarized cells, but A549 is from the lung epithelial. If you think about the 3D context, it can be polarized. So polarity factor is there, but the system I'm using is a 2D culture system. May not necessarily tell that it is a polarized cells.
And also, just very briefly, the slides you had about the stimulated depletion microscopy.
Correct.
I'm not familiar with that technique. Can you just very briefly?
Yes. Yeah. The STED technology is the high resolution imaging because there is when you're talking about the confocal microscopy, there is a light diffraction limit. We cannot go more than 200 nanometer scale. But the STED is advanced, more sophistication in a way that they use the two pair lasers. One laser they excite, example you excited 488, and another laser beam will come as a donut shape. What it does that, okay, you excite and have a donut shape, and then you deplete all the excitation diffraction light. Then you have a very circular, thinner. Then from that 200 diffraction limit, you can go below, I think I would say 70 or 80 nanometer scales, if I'm not mistaken. So that allows the resolution. That's why we can able to see virus filament, and it is an individual filament rather than a some kind of just fluorescence. Yes.
So how do you transfect the other, like the first cell and the second one? Like you have transfected cells or infected cells, right? And then put those cells into another dish of cells?
In this experiment?
Yeah.
No, these all cells are infected in the same place, like meaning that for the siRNA transfection, we used a reverse transfection method. We put the siRNA and then we put the cells. And we allow 48 hours to have the knockdown achieved. And it has been trial error many times, and, and we are confident that 48 hours time point, more than 90% protein was reduced. And those cells infected with RSV GFP, so we are not transferring that. So we're using MYF1, and in this case, we're 0.1, because allowing 1 day, then we can identify a green cells already there. So then we can focus the microscope in that cells, and then imaged over time, and these images is 3 independent spaces. And we also did multiple independent times. So that's why the movies are from 24 hours to almost another 24 hours, and we imaged in the same area. And when we did, we have a technical replication as well.
You didn't claim that the viruses in one cell are being transferred to the other cell through these
Filopodia. I did claim, I hope you will appreciate for the state imaging we were able to show that.
So my question kind of is that how do you know that the viruses are not in the dish?
Yes. Yes, what we know so far that virus always shed from the infected cell surface in the supernatant, and then you always reach out. But this is the first time we are able to show that. And to convince, at least we are, definitely we need to do more experiment. But to our, based on whatever findings so far, we are able to see Filopodia in the confocal. We did more resolution that we actually see the virion by state. We using capturing that movie based over time. This is actual movie. This is not imaged over time, and they just combined it. And we were able to see that ARP2 knockdown impact that process. So we are close enough to confidently say that this is the first time a negative strand RNA virus, Filopodia, is used to cell-to-cell spread. And for ARP2 knockdown, if you can see that Filopodia is not much as control. And the infected cell to spread in other cells is also not as efficient as like siRNA control. So it also gives the idea that ARP2 is involved in this process. One of the caveats in this experiment is we used a semi-confluent monolayer. But in our human body, all cells are stuck together. But we cannot infect ourselves. Rather, I used a confluent monolayer to recapitulate this phenotype. And for that, I used similar rate-affecting cells. And regardless of confluency, you will see the infected cells is more dynamic, and they can make syncytium as well. And then cytopathic effect, allowing the space, then infected cells can come and taking that space, and more, you can see how dynamic it can be to try to reach out to other cells. So the virus infection is really taking advantage of the that area of the cells. And if ARP2 is involved in that process, we expect that the monolayer can be retained much longer. Exactly, we found that ARP2 knockdown, even though these are infected, the spread was not obvious as like siRNA control, and the monolayer retained much longer. So what it suggests that if we can able to have come up with a therapy in the lung, and that will allow longer time that immune cells to kick in to combat the RSV infections. That's quite amazing in our results. And when you're talking about, do you have a question? Yes.
So does the ARP2 knockdown affect cell viability normally with just the knockdown, or is it that it's just affecting cell viability when you put the virus on?
Yes, to answer your question that ARP2 knockdown itself reduce the cell viability or not, answer is no. But that was the first question before even this study. I found a good siRNA that 90% protein reduction. I beg your pardon that I did not use that in that slide because of the time I take it out. And the viability is no difference, like the above 90% viability. I also check because I also make a constitutively ARP2 knockdown cells, and the constitutively ARP2 knockdown cells grow as efficiently as FF49 cell. In some cases, even better. So that's something, another point of investigation, about how better or not. But based on my experimental results, I can tell you that ARP2 knockdown did not reduce viability, and constitutively knocking down ARP2 FF49 cells grow as efficiently as wild-type cells.
But when you have the knockdown and then also infect with the virus,
Yes.
it does affect viability.
that viability effect is due to the virus-driven infection. What we found so far is that virus can infect cells. ARP2 knockdown impact the virus spread out. So it's spread out when I'm talking about in the twofold. A, the budding process is reduced. B, cell-to-cell spread is reduced. Because I'm not showing here the RSV also making the syncytium, and syncytium did not impact it. And kind of makes sense that because syncytium is due to RSV F protein. So if all cells are together, and then F can easily making them syncytium. But in respect to the spread, when we're talking about the shedding through budding or cell-to-cell is the novel mechanism, in both ways we have impacted. And that's why we see the tenfold reduction. Tenfold reduction may sound very is a modest, but if you think about the RSV grows very low titer compared to other viruses, therefore tenfold is insignificant in some cases. And so as I showed that cells are migrating, so I wanted to quantify or making a migration assay that allowed to identify the inhibitors. So traditional migration assay, what we do that cells will crawling up and we will measure the distance. But hope you appreciate that RSV infection makes cells dynamic, so it can migrate everywhere. So therefore, I have to tweak the experiment like this is the red affecting cells, and if cells migrate in the scratch area, we'll quantify the red signals and we'll normalize with the overall field, rather than making the distance of linear distance. And infected cells, we expect that it will be more migration. If F49 cells is inherently migratory, you can see the cells are coming in that area. And our expectation is if RSV induced Filopodia and Filopodia involved in the migratory pathway, then we expect that higher cells will be migrated in the scratch wound. And we exactly find that. And for that, you can see the blue line, RSV infection or cells migrate over time, and ARP2 knockdown, this migration reduced substantially. And you can see cells inherent migration over time, and ARP2 knockdown without infection, they do reduce the migrate as well. So then now we have an assay to identify whether we can inhibitors that will reduce the RSV-driven cell migration. And exactly we found that we used first CK666 is a potential inhibitor for ARP23 complex. And I also used inactive control 689. And as you can see, the blue line showing that RSV migration as expected. And we used 689 as an inactive control. And inactive control did not reduce migration that much. But if you see about CK666, the migration is substantial. We also used another inhibitor that also targeting ARP23 complex, basically target ARP3 protein, and we see higher reduction in here. And without virus infection, like CK666 and 548, both are reduced migration. So it's confirming that we can use a migration assay to identify inhibitors that can be used a therapeutic target. So, but problem is CK666 has not been used in animal study. We do not know the bioavailability or PK/PD value. But encouraging thing is that there is a drug, it's called CDD methylesterase. It's a synthetic triterpenoid drug and has been FDA approved and has been long using for chronic diseases as well as cancer. And researchers have shown that this drug is target ARP3 complex. So currently I'm doing now identifying a good concentration that reduce the RSV driven migration, and hope that this drug I can test in the animal model and in vitro model to hoping that that drug can be used in the therapeutic purposes for an RSV infection. So that is quite encouraging that we were able to find a cellular phenotype, cellular factors, and targeting a drug to start with. It's a long way to go, but there is a hope.
What are some of the animal models for RSV?
It's a very good question. And there is, it's not a good animal model. There is no good animal model for RSV. The only model that recapitulate the lung pathology is by the cotton rat model. Cotton rat, yes. mice is they are permissive, but not lung physiological aspect is not a good model. Even not hamster, they can grow upper respirator, but the lung physiology or the RSV-driven lung pathology is only can be recapitulated in the cotton rat model. Yeah, and I'm planning to do some studies with the CDD or methylesterase in the cotton rat model. And so I showed you RSV. So another question is whether is this RSV specific or not, this phenotype. And I used the two relevant viruses. Human parainfluenza virus belongs to the Paramyxoviridae, and metapneumovirus. And neither of these virus infection induce Filopodia. And we confirmed with when we quantified the Filopodia and also imaged it over time. So our results suggest that Filopodia is RSV specific. And we found that RSV specific, we want to identify whether viral factor is involved in this process. And to know that, we use RSBF transfection, and we do see Filopodia-like structure, but not as efficient like virus infection. But in compared to control, we do see Filopodia-like structure. Not only plasmid, we also use a viral vector system. And we found that, I showed in the previous slide, HPIV3 did not show Filopodial induction. But we introduced RSBF protein in that virus backbone, and we found that Filopodia can be introduced, but less in number. And these Filopodia induction can be impaired by when ARP2 knockdown cells used to infect those cells with this virus. So confirming that RSBF has a role in the Filopodia induction. And so here is the model, which looks like that. We found RSBF protein and the cellular protein in the Filopodia induction. And these results was highly appreciated by the PLOS-PAT. They make a cover issue our results. And you can see the nice image how virus infection induced Filopodia, and this Filopodia used for transmitting virus. So it's quite amazing in respect to virus can efficiently hijack the cellular machinery for their own benefit. So as you can imagine, that whether any other upstream regulator are in this process. Exactly, and we found that this Filopodia or Lamellipodia, there is a row GTPase is involved in the upstream. Like example, CDC42 involved in the Filopodia, whereas RAC involved in the Lamellipodia production, and Rho is the stress fiber. And if we go into further, that CDC42 use NWASP for actin polymerization. What I did, we did exactly the same study as I showed you. Because of the time, I'm not going to any of the data. But what you find that knocking down CDC42 or NWASP, we see reduce in the Filopodia, reduce in the virus production, and also reduce in the migration. The result I only show that here the control RSV infection induced virus migration, and if you knock down either of these protein, reduce this RSV-driven migration, confirming that we found upstream regulator for this process as well. Probably you also asked, yes?
Were those targets identified on your, were those genes identified as targets on your original screen?
That's a good question. So we were very stringent when we are looking for the target. So we not only used stringent criteria, so therefore in the first 65 was not in there. And there are a couple of reasons could be, because when we're talking about high throughput screening, and those are basically off-target effect, and some other drawback limited to identify those factors.
Did they mimic the effect on viral titer and virus protein reduction?
Yes, and even a little bit bigger. There the fold I showed you 10-fold, and CDC42 more than 10-fold. And it could be because it's in upstream, and maybe some other factors is involved in this process. But what it tells that that it could be a potential upstream regulator based on that Filopodia as well as the virus production reduction due to knockdown. And probably also wondering, what about upstream of CDC42? And interleukin signaling, it could be one possibility, because my project has started with interleukin signaling. So, so therefore, I wanted to investigate further whether the 4 of the protein I talked about, to start with, whether impacting this RSV production. And first, I showed you that we have a siRNA successfully knocking down that protein, but when we check the viability, integrin showed reduction in the later time point. And as you can see, there is a later time point, but when we checked the early time point, the entry process, I beg your pardon. Early process, you can see that ADP ribonucleation factor 1 and integrin alpha 5 has the impact on RSV entry process, but neither ARP2 nor MAP kinase is involved in the entry process. To answer that question, that MAP kinase is still not involved in the entry process, like ARP2. But what we found that MAP kinase is also involved in the RSV-driven migration process. And if it is true, then we also look for the total virus production. And for that, when we quantified that, you can see ADP ribonucleation factor 1 and integrin alpha showed the highest reduction, but there is an indication of the MAP kinase not necessarily as high as ADP ribonucleation factor 1. And we also recapitulate the result in the CALU3 cells. So there is an indication that integrin signaling may be involved in the RSV infection process. So with that, I will, to summarize that, what I found so far is I identified a genome-wide siRNA screening that allows me to indicate that integrin signaling is associated with RSV infection. What I found that RSV infection not only induce Filopodia, but also increase cell migration. And we found Filopodia as a novel mechanisms, ARP2 is a novel host factors, and CDC42 upstream of that regulator, and we also find a viral factor, a protein, more likely involved in this process. So here, and we have a model for that maybe virus hijack this CDC42-driven Filopodia induction pathway for its own benefit. So I also want to expand my research is that I talked about CDC42, but there are many other Rho GTPases, like example Rho and RAC1, and they also involved in some part of cytoskeletonic, example microtubule or some other cases. So I want to delineate that whether Rho GTPases contribute independent of ARP2/3 or not in the RSV process. I also want to know whether there is any crosstalk between them, because all are cellular cytoskeletonic pathway utilized, whether RSV used specifically CDC42 or it Rho1 or RAC1 involved in this process. And if I find able to example CDC42 involved, then I want to recapitulate that in the primary cells, because all I've been doing is the 2D culture system. I want to go in a 3D culture system that allows me to more recapitulate the results in the primary cells. And these findings definitely going to be very helpful to understand whether RSV-induced bacterial co-infection. I did not talk about that, but there is a possibility that viral infection allows commensal bacteria to be invasive or more infectious. And so if we know that these pathways, we're probably able to identify how bacteria take advantage in the RSV-induced or any other respiratory virus-induced pathophysiological aspect. And we also can recapitulate or can study this system in other virus replicative cycle as well. And we know that cancer metastasis involve Filopodia, because 90% cancer death is because of that cancer or metastasis cells' ability to invasive to the organ and then organ failure. So if we know how the Filopodia or some other cytoskeleton structure involved in this process, probably we can find a potential anti-cancer drug as well. And definitely neuronal growth cone and dendrites use Filopodia or ARP2/3 complex-driven actin polymerization pathway. So this study hopefully can collaborate or giving some positive direction in this research as well. My second aspect that I alluded a little bit about the cell type specificity. Here is a Vero cells. Hope you'll appreciate that. Vero cell, even though it is epithelial cells, but we do not see a Filopodial induction much. But if you see one more time how RSV is Filopodia induction and making syncytium, I hope you'll appreciate this completely, there possibility of cell type specificity involved in this process. So I wanted to further investigate to establish whether a cell type specificity involved, if there is no cell type specificity, whether the tissue specificity involved or not, or whether species specificity or not. And if I can establish that there is a cell type specificity, then I want to determine the cellular factors. For that, I can use microarray or even viral protein-driven immunoprecipitation-based pull down, and then do the mass spectrometry to identify the cellular factors. And talking about cellular factors, we can also identify the viral factor as well. I just showed you RSVF. What about NS1, NS2, or non-structural protein may involve in this process as well, because there are some other viruses uses non-structural protein for actin-related pathways. So there is a lot of we can do. We can use a pyrene assay, that is the extracellular assay. You can identify whether purified protein initiate actin polymerization or not. So this finding definitely will identify novel targets for antiviral, not only for RSV, but also other negative Western RNA viruses that more likely involved in this process. And another thing, interferon signaling, I showed you some indication there is a possibility involved in this infection process, but I wanted to characterize comprehensively that whether these proteins are involved. And another point is that how interferon, if interferon is involved, how will be the mechanistical point of view? Because we know that in the lamellipodia, the interferon allows make a molecular clutch where give the strength of the cells to protrude like a Filopodia. And this molecular clutch is involved in this focal adhesion, and focal adhesion involve almost 80 proteins involved in this process. Another important protein is the focal adhesion kinases. And these kinases allow to transmit signals, Rho GTPases. I showed you the CDC42 Filopodia and the cell migration. So there is a Pandora box there. We can investigate further to identify how virus efficiently hijack cellular migratory pathway for its own benefit. And definitely, if we can identify that, and these results can be utilized to understand bacterial co-infection, not only that, but also we can identify a novel target for antiviral as well as antibacterial. I will wrap up, finish my talk to acknowledging I've been fortunate enough to supervise by great mentors like Peter Collins, and all the members in his lab is very generous helping me to do this project. I'm also thankful to Scott at the NCATS as a collaboration to did the screening for us. And I am very grateful to two persons in the biological imaging system, Marjorie and Shundar. They helped me confocal state microscopy as well as developing that migration assay. And I also thank to Elizabeth Fisher who are helping me for the electron microscopy. The last but not the least, I'm also thankful to my family giving me support all the time. And I will leave with you a model that you can see how Filopodia can transmit virion on the cells, how amazing this is that virus can take advantage. And I hope give you a model, and happy to answer any questions you have.
Time for a couple quick questions.
I have two quick questions. I don't know if these
Or
are
maybe we'll do
couple
one
you're
more
thinking
after
about.
that. Yeah, go ahead.
So in your model of Filopodia, it's very interesting, right? But I usually think of some of these mechanisms more along the lines of penocytosis and viral entry and exit. Wouldn't that be a more obvious reason for differences that you're seeing here? Would be mechanism associated with viral entry and viral exit rather than this Filopodia?
Yeah, that's a very good question, that whether because Filopodia may be in the entry process, like the macropenocytosis has been suggested that RSV entry process, but macropenocytosis signaling, if you think about that, at least in our result, ARP2 is not involved in this process. And macropenocytosis, if
Did you directly look at that?
because based on the ARP2, like the ARP23 is not impacted, because if you see, remember, EIPA is a macropenocytosis inhibitor that reduced that. But what I'm point of view that signaling cascade that I am investigating is the ARP2-3 related, based on our findings so far. And what we know based on our findings that ARP2, 3, using the CDC42 is more necessarily involved, and that is more likely Filopodia and the spread aspect of it. But in respect to the entry, whatever endocytosis, maybe there are many other CDC, like CDC40, Rho1 involved in the endocytosis, or no, exocytosis, vesicle transport. RAC1 is involved maybe clathrin independent. So there is a diversification of the Rho GDPases dependent signaling cascade. Maybe there is a crosstalk that I want to investigate further, but to the best of our knowledge, based on our understanding, is that ARP2-3 driven initiation, primarily CDC42, is a master regulator, and CDC42 has been known that is a master regulator for cell migration on Filopodia. And in what we found so far, that RSV used for the spread. The entry, at least in our result, not necessarily this pathway.
So my other question deals with the system, right? So you're using A549s.
Right.
You're also using the cow, the other.
The monkey kidney cell, verocells.
So all of those are non-ciliated, right? So,
No.
So physiologically speaking, RSV prefers to entry and exit ciliated cells.
Right.
When you expand this to a 3D culture or something more physiologically relevant, how reflective do you think your data are actually going to be of the more physiologically relevant situation, right? I mean, is all of this just A549s, or do you expect to see similar back?
Yeah. That's a very excellent question, and that is the most important question. If we think about the physiological ciliated cells, cilia is primarily microtubulin on the top, but at the edge of the basis is the effectin, the Filopodia, when you're talking about Filopodia, is extensive of the effectin there. So in respect to
Those cells are polarized, right? So
Yeah.
you can't really say which side or where they're coming from.
Oh, in the 3D culture context, in the epithelial layer will be, and has been shown that RSV use that cilia to get in, and but it does not go to the basal layer as well. So the point is how I can relate to that process. One of the point is that even though if we think about very theoretical aspect of it, okay, if cilia is a microtubulin, where is talking about effectin or the Filopodia-like thing. So when we're talking about effectin, there is a ciliated base is based on the effectin. Now, in respect to the transportation of the virus, the microtubulin also used for the intravesical transport, but effectin also can be involved in the transportation process, because effectin allows to give the base for the cellular transport and some other extension. What we are talking about here, how effectin produced. We're talking about ARP2-3 complex-driven polymerization. We're talking about how this actin polymerization signals comes from. We're talking about CDC42. And now what we want to know, how virus use that canonical thing for their own benefit. So in respect to the physiological context of it, I do not have the data right now. I am doing hands-on, doing the primary cells using that epi-array culture to trying to image where the virus and where effectin and where is the microtubulin in the 3 culture systems. So I wish I had that results to tell you, but there is a hope because effectin is there, and if virus utilize the signaling pathway, we can expect there is an involvement in this process.
I brought it up because 2 of the 5 pathways that IPA analysis picked up
Yeah.
are associated with bladder cancer and ovarian cancer, right?
Yeah.
That's probably just background noise.
Right. Right. And because IPA is a knowledge base, they give a probability of how likely already shown the protein is involved.
Genes match the other.
Right.
So but that tells me that most of those genes are probably background noise from the A549, which could also be why you came up with the kinase.
Right. There is a possibility always, but IPA allows to give an indication that gives a probability to look for, because when we're talking about the screening, it's so many genes to work with. We need to have a way to narrow down, to focus, and try to go for it. So that's what I did, rather than cherry picking and identify that.
Other questions? Okay, if you would like to hear more about Dr. Mehedi's future research goals, he will be giving a chalk talk tomorrow in room 1059 at 3 o'clock. So I encourage all of you to attend. Okay. All right, thank you.
Thank you very much.